Hello, my name is Karina and I am the Scientific Marketing Manager of Savoy. We are the original manufacturer of cellQART inserts and trackage membranes for off-the-shelf and customized cellQART applications. I am very happy to have the opportunity to present in the 11th World Congress on Alternatives and Animal Use in the Life Science, and I would like to thank the organizing committee for this invitation. Having more than 60 years of experience in the manufacturing and commercialization of life science products and NAMs, we would like to share with you our own experience and provide a guide for an effective product roadmap towards large-scale production. Our goal today is to create awareness regarding the role of manufacturers and suppliers towards the 3Rs principle. We are delighted to be a sponsor of these events as we are strongly committed to the 3Rs principle of refining, reducing, and replacing the use of animals in research and testing. Savoy is actively working to accelerate the in vivo to in vitro transition. Our tissue culture treated trackage membranes are excellent support for cell growth and can be found in cell culture products and state-of-the-art on-a-chip systems. In addition, in 2020, we launched our home brand of cellQART cell culture inserts, making highest quality cell culture inserts available at affordable prices. We would like to start this presentation with some facts about animal testing. In vivo testing has been the gold standard for preclinical drug development and toxicology. However, animal-based methods lack human relevance and have a low predictability rate, not to mention the enormous ethical and financial concerns associated to their use. For instance, the average time to take a compound from the preclinical phase to the market is 10 to 15 years, with a cost of approximately $2.5 billion. The fact that 90% of drugs tested in animals and entering clinical trials fail due to lack of safety and efficacy questions the significance of in vivo testing. In addition, only 25% of the toxic effects observed in animals are expected to occur in humans. However, the field is progressively transitioning to alternative methods. For example, the European Union Reference Laboratory on Alternative to Animal Testing has listed 365 alternative methods, and currently there are 49 new approach methodologies that are included in OECD test guidelines. In 2013, the European Union banned the marketing of cosmetics containing ingredients tested on animals, and other regions have since followed. Several key driving factors are involved in this growing transition. Generally speaking, in vivo methods are very costly and time-consuming, especially when considering long-term exposure studies. In addition, they are technically challenging and impose many ethical concerns. Therefore, the high-throughput potential is limited, and the control of the chemical and physical environment is low. That makes it really difficult to draw accurate conclusions. Apart from those aspects, one key factor that questions the pertinence of the use of in vivo methods is their relevance to human health. The poor predictability of animal experiments is one of the biggest issues in the field. The use of non-human animal species and different strains with a variety of metabolic pathways leads to variation in efficacy and toxicity, making this cross-species extrapolation an important issue. In addition, animal models often do not accurately predict adverse reactions or recapitulate important aspects of disease in humans. On the other hand, in vitro methods allow the use of human cells, eliminating the issue of cross-species variability, and can help reveal mechanistic information as to how a substance may exert biological effects. That provides human-relevant insights to make more accurate predictions. In addition, their scalability and high-throughput potential increase reproducibility and statistical power. Despite the many advances in the in vitro field, some challenges toward the implementation of new approved methods remain. Apart from scientific excellence and human relevance, new methods should empower a regulatory and industry paradigm shift in comparison with animal testing. Supplier industries and manufacturers are critical stakeholders to achieve this transition, and their inclusion is key to make new technologies available. In this regard, the supplier industry has been a driving force towards the transition to non-animal research by translating new approach methodologies into scalable products that have been adopted by the regulatory and testing industries. For instance, The supplier industry has been actively involved in the promotion of the use of NAMs, and there are many examples towards the development and implementation of new in vitro methods. Several NAMs have already been successfully adopted in the early steps of testing as predictive tools for informed decision-making. For example, according to a recent report, in vitro models of the gut epithelium, lung and eye are currently included in the drug discovery pipeline of relevant pharmaceutical stakeholders. For preclinical safety studies, models emulating liver, bone marrow, gut epithelium, lung, and skin are established. NAMs mimicking the liver, lung, and kidney are being used to evaluate pharmacology and toxicology effects, whereas vasculature, liver, and pancreas models are utilized for target identification and validation. The supplier and manufacturing industry are paving the ways towards the regulatory approval of alternative methods as well. For example, the FDA has signed a research and development agreement to use NAMs to study and evaluate the efficacy and toxicity of regulated drugs, including COVID-19 vaccines, using, for example, brain in a chip, intestine in a chip, liver in a chip, and lung in a chip models. Furthermore, a consortium between the NIH, the FDA, and 22 companies has been formed with the intention to provide an integrated voice in the field of microphysiological system for drug testing. In an unprecedented event, and as a result of a decade-long collaboration between industrial partners, the world's first animal-free toxicology strategy was approved by the OECD. This strategy combines 3 NAMs to assess human skin sensitization, and it is better to predict allergy risk than animal testing. This is a huge step towards the validation of NAMs, and this advance could have not been possible without the involvement of the industry and the cooperation with scientists. Examples like these are strongly encouraging and show a robust commitment from the supplier, pharmaceutical, and testing industries to science, human, health, and animal protection. The implementation of NAMs requires education and dissemination efforts as well. Among the goals of those didactic approaches are to help facilitate the wider adoption of new in vitro methods by providing information on the available alternatives and to communicate scientific facts to build confidence as efficient tools for education, research, drug development, and testing. The supplying industry is playing a key role in educating potential users through the generation of easy-to-access content and resources directed on the selection and setup of the correct NAMs application. The mechanisms used to promote the 3 Rs involve staff and training opportunities, courses, scientific seminars and meetings, awards, communications, newsletters, discussion panels, audiovisual content, among others. These educational resources are particularly useful to select the right non-animal assay within context of use and applicability domain, and also to share experiences between companies to learn from each other. The strong investment of the industrial stakeholders in the generation of transparent educative resources reflects the commitment on fostering the development and application of new approach methods. Whereas scientific advances in the NAMs arena have spiked in the last decade, there are many commercial hurdles that are necessary to address to make from this alternative approach a new sustainable standard. From our 60 years of experience as plastic and membrane OEM of commercial off-the-shelf and customized life science products, we would like to highlight some of the main challenges, and we would like to provide a piece of advice on how to transform those challenges into opportunities. Our goal is to raise awareness on the status from the supplier industry point of view and to open a forum of discussion and mutual collaboration towards the implementation of new in vitro approaches. One of the main challenges faced in the industrial production and efficient commercialization of NAMs is the need to improve the communication between end users, developers, and suppliers, not only during the final discussions, but throughout all the process. This is a critical step required to identify the real unmet needs and to assess the viability of the propositions, as well as to set up realistic expectations and limitations. This clear approach assures the proper evaluation of the product and prevents the misuse of resources, where there is no significant added benefit or demand. From our experience translating NAMs into commercially available products, lack of communication results in high cost and long time to bring a concept to the market. We believe that this is caused by two main factors, which may appear trivial but are essential elements that should not be overlooked when translating ideas into commercial device. One is the underestimation of product requirements and defined intended use. And the second is the inability to reproduce low-volume manufacturing results in a larger scale. In the following slides, we will provide some useful information to avoid those problems. A list of obvious prerequisites focusing on the core application is what most inventors start with. However, unnecessary project iterations can arise when the functional and non-functional requirements are not adequately defined and integrated into the product. This is especially the case when approaching an industrial injection mold or assembly machine supplier to manufacture the initial industrial prototypes. Beyond dimensional parameters, the following specification elements should be pulled in during the early definition phase. One is the material selection and tolerance levels to comply with biological evaluation, according to applicable standards. The second one, and very important one, is to define the regulatory strategy to follow. Another element are the environmental conditions, including packaging, labeling, and transport profile. In addition, all critical quality parameters affecting homogeneity of cell growth to enable reproducible results should be considered. The quality control and product acceptance levels are also important elements. And finally, it is important to consider the objective measures to prove successful verification of all these requirements. While lab results may have shown sufficient proof of concept, the final acceptance is created by the ability to generate reproducible results. This is strongly related to the aspect of complete requirement definition as a precondition. A highly integrated solution provider should guide the inventor through the potential design options with affordable prototypes and develop final specification parameters in an agile way. Once the final stipulation has been released, the assembly machinery and injection molds can be built. Unlike requirements characterizing the product, during the process validation, the final production parameters are defined as a result of a design of experiments. Only after a successful validation process, manufacturing ramp-up can start. The finally selected supply source should be able to demonstrate a strong reputation for each of the single components and the assembly of these into a final product to ensure the highest quality even at larger volumes of production. NAMs should be developed from the beginning, having in mind not only the human physiological relevance, but the potential market size, scalability, reproducibility, and high throughput potential. A well-balanced interaction of ergonomics, appearance, functionality, technology, and ecology are very important aspects as well. Having all those parameters in mind is of fundamental importance to develop a robust business case and an effective product roadmap. Many NAM devices contain plastic components and semi-permeable membranes that serve as support for cell growth. Therefore, it's important to select the right manufacturer. This will aid the regulatory acceptance process, propelling the adoption of NAMs and paving the road towards a world with new standards. In close dialogue with our customers, we develop specific plastic molding solutions and membranes that are PFOA-free for future products. We bring innovative and high-quality products to market acceptance, always in compliance with the regulatory provisions. In our state-of-the-art mold manufacturing division, we're building new injection molding tools. Ultra-precise tools for the injection molding process are the basis for a reproducible production of complex components in large quantities. As an OEM supplier, we produce in accordance with the requirements of the GMP and ISO 13485 in control areas or clean rooms, ISO Class 8. Savoy develops and manufactures its own microporous tissue culture treated membranes. For the assembly of membranes, Savoy creates tailor-made production designs based on specifications. We guarantee a high availability and a secure economic production of the products in clean rooms and control areas. We are unique as all the production of our plastic and membrane components, their assembly and packaging happens under the same roof. This allows us to have a strict control and we guarantee 100% parameter consistency. In addition, we are familiar with all common sterilization methods and will help you select the suitable method for your product. As you can see, a systematically designed, modular research and development process creates transparency and allows us to step in during any product development phase. Now, I would like to talk to you about one of my favorite products, which is also a successful example of such development. And this is our cellQART inserts. Our inserts are ideal to grow cells in an in vivo-like environment, as the semi-permeable membrane allows access to nutrients from both the apical and basolateral side, encouraging the formation of a tight cellular layer, improved morphology, and also differentiation. With the inserts, it is possible to model tissue barrier models, perform co-cultures, study the effect of secreted molecules, perform migration and invasion assays, air-liquid interface cultures, among others. Our inserts are currently used by academic labs all over the world, as well as contract research organizations, cosmetic centers, and toxicology facilities. They have a convenient pipette access, an optimized gas exchange by product design. They are RNase and DNase free and non-pyrogenic. As I mentioned before, we guarantee 100 membrane parameter consistency and reproducible cell culture results, and they are produced in an ISO Class 8 clean room. We feel proud to contribute to reducing the number of animals used for scientific procedures, and we will keep working towards this goal, and we will keep setting new standards based on ethics and quality. Our trackage membranes can be found in many state-of-the-art on-a-chip models, helping advance the quest towards innovative, animal-free approach methods. And we look forward to continue contributing to the development of new research and tested platforms. Finally, we would like to conclude with the following remarks. Early engagement with established manufacturers and industrial partners brings support to NAM developers to translate their application needs into commercial, technical, and regulatory requirements towards larger-scale production. In this regard, Savoy offers off-the-shelf cell culture inserts and project-based customization with 100% in-house production of plastics and membrane components. Savoy is committed to help to enable the development of reliable in vitro cell-based methods with the goal to refine, reduce, and replace animals used in research and testing. By partnering, we could progress to develop more powerful, reliable, and financially self-sustainable methods that mimic the complexity of higher organisms. Finally, we believe the goal to reduce, refine, and replace animal testing is as important for the animals as it is for us. By developing standardized, human-relevant NAMs of industrial interest, it will be possible to improve the assessment of chemicals and cosmetics, reduce drug development failure rate, advance human benefit, and protect animal welfare. I would like to thank you for your interest in this presentation. We would love to have a conversation with you. We would like you to test our products. Please follow us on YouTube and Savoy. In Twitter, we are as cellQART. Please feel free to contact us also to the emails that are listed in this slide. And please share your thoughts and let's make this new approach method the new standard. I wish you an amazing rest of the day. Thank you.